So any questions I answer would be in it. Gonna wait a couple more minutes for a few more people to jump in. But yes, know the different steps between translation and transduction. Yeah, but more so know how they're different. And know the different kinds of RNA that are involved in translation. And it's translation and transcription that are the two that go together. So can anyone tell me what transcription is? You can type in the chat or unmute yourselves.
Isn't it making RNA from DNA?
Yes, yes. So okay. Transcription is DNA from RNA. And then translation is RNA to protein. Yes. And then transduction is a slightly different thing. Everything in biology sounds the same, like they all start with the same roots. So it can get confusing. But translation and transcription go together. And then transduction is a type of genetic transfer. Does anyone know? So it's transduction, transformation, and conjugation, which are the types of genetic transfer. Does anyone know what is a defining factor of transduction genetic transfer? Yes, yes, it does require a bacteriophage. All right, it is 3:53. Meeting a few more people. Transduction. Yes, transduction is the transfer of genetic material using a bacteriophage. So when a bacteriophage, a virus, infects a bacterial cell, and it, in turn, when the virus replicates, carries some of the bacteria's genetic information over to other bacterial cells. There are a couple of different ways it can do that within transduction, and you need to know those as well. Like the difference between generalized transduction and specialized transduction. But yes, transduction in general requires a bacteriophage, while conjugation and transformation do not. Does anyone know what is required for genetic transfer in conjugation?
Isn't it the conjugation pilus?
Yep, it is the sex pilus, also F plasmid. Yes, those are also a thing that happen in conjugation. The F plasmid is what is being transferred back and forth, or the F plasmid is what can make a sex pilus. Yes. All right, I am going to, hello. Okay, now I'm going to get started on the Kahoot. Can post the Kahoot game so we can play it ourselves too. Yes. And I will also put all of the questions that are on the Kahoot in a like study guide-ish thing, like without the answers, so you can go through them on your own. I'll also try to figure out a way to, yeah, I can also try to post the Kahoot. That might actually be easier. So here, let me start that now. Okay. And I'll share my screen with you guys too. Okay. And I made it a Halloween Kahoot because it is Halloween season, and I thought it was a lot more fun, and we can have fun while studying. So share screen. Okay, and share. All right, so go to kahoot.it and enter that game pin, and once you guys are all in, we can get started. Double checking. Oops. Okay, just a few more people. Are we all in? All right. I think so. Mhm. You got kicked? Here. Oh, did you got back in? Awesome. All right. For anyone who just entered, we are starting the Kahoot very soon. So sign into kahoot.it and put in this game pin and we will get started. Just one more minute. All right. Okay, I'm going to start this now, and then I think anyone who joins a little bit later on can also. Oh, are we all in? Yes. Okay, and starting. All right, and this has questions kind of guided by the study guide that Dr. Kokesh gave you guys, and also some questions just from the material in general. All right, what is the central dogma of genetic information? Yes, very, very good. It is DNA to RNA to protein. So yeah, make sure you're reading the question. That is a fairly easy one. It always goes DNA to RNA to protein. It's transcription, translation. Next question. Awesome job, guys. All right, true or false? Phenotype defines the genetic makeup of a cell or organism. Very good. So a bit of a tricky one. Genotype is the genetic makeup of a cell or organism. Phenotype is the physical traits that something has based on the genotype. So phenotype is based on genotype. Genotype is the genetic makeup itself. All right, next question. What is the complementary mRNA sequence to this DNA strand? I don't know if I put enough time for that one, but yes. So put that orange one in there. It's a little bit tricky. Good job not doing the red or blue. If you are asking for an RNA strand, if something's asking for an RNA strand, you can immediately eliminate anything that has a T in it. There is no thymine in RNA strands. It's uracil. Hi, we're just playing the Kahoot. I think the game pin is at the bottom if you would like to join. But next question. Yeah, probably should have provided a bit more time for that one. Takes a little bit to go back and forth. What does it mean for DNA replication to be semi-conservative? Yes, a new DNA strand is made of one parent strand and one daughter strand. Very good job. A new DNA strand is not two daughter strands. So if you think of it, so like you have a DNA helix, it unwinds, and then in between them, a new one is formed. And so two new DNA strands will appear with one parent strand and one daughter strand, and then one parent and daughter strand in the other one as well. So semi-conservative means one parent strand is kept. What kind of RNA carries individual amino acids to the ribosomes for making new proteins? Important to know your different types of RNA. Very, very good job. Awesome job, everyone. I always got these mixed up, but you guys seem to know it very, very well. So awesome. Yep, it is tRNA. mRNA is the one that is transcribed from the DNA directly. Transcription is DNA to tRNA. Very good. Bit of a trick question there. So yes, that would be false because transcription is DNA to mRNA. Where does translation occur in prokaryotes? Translation. In the cytoplasm. Yep. Very good. If someone answered mitochondria, it is a prokaryote, does not have any organelles. So cytoplasm is where translation occurs in prokaryotes. Okay. Which bacterial DNA transfer type requires a bacteriophage? I went over this one a little bit earlier. But all the words sound very similar, so make sure you're reading the questions. Transduction, yes. It would be transduction. Translation is not a type of DNA transfer. It is DNA to, it's RNA to proteins. Transformation is the other type of, transformation and conjugation are the other types of DNA transfer. Next. Okay. What is a characteristic of specialized transduction? So yes, it is the red one. A phage incorporates itself into the host chromosome. That is specialized transduction. Generalized transduction is the blue one. So glad none of you guys answered that one. But a plasmid assisting in transferring bacterial DNA from one cell to another, that is conjugation. A plasmid is used in conjugation. A bacteriophage is used in transduction. All right. True or false? Transformation is bacterial DNA transfer that requires cell-to-cell contact via a pili. False. Very good. So yes, transformation is naked DNA transfer from cell to cell. There's no bacteriophage involved. There's no pili involved. It's just naked, like free-floating DNA transfer. Conjugation is the one that requires cell-to-cell contact via a pili. All right. Which of the following is not a type of mutation? Yes, very, very good job. Missense, silent, and nonsense mutations are all types of mutations. Nonsense mutation doesn't sound like it should be one because, I don't know, it's just a funny name for something that's actually a thing in science. But yes, a nonsense mutation is real. A nonsense mutation is changes result in a stop codon, but repair mutations, not a thing. What does it mean that the genetic code is degenerate? Some codons are redundant, so several codons code for the same amino acids. Very, very good job, guys. chain conjugation, the cell that is receiving the F factor is which one? Yes, the one that is receiving is F negative. It doesn't have an F factor, so it is F negative is how I always remembered it. If it already has an F factor, meaning it is able to give it, it is F positive. Next. All right, on to bacteria. Rickettsia, Wolbachia, and Bordetella are all which? Awesome job, guys. Yep, they are gram-negative rods. And if you are in the Zoom, but you're not in the Kahoot, you are more than welcome to go to kahoot.it website. And then if you can see the game pin in the bottom of the screen here, it is 4916637. You are more than welcome to join. All right. What bacteria secretes a blue-green pigment into the surrounding media? It is Pseudomonas aeruginosa. Very good job, guys. You will need to know some fun facts about some of the bacteria on the study guide. What I did is, so for bacteria, know how they stain, know their shape, know a fun fact about them, and know their oxygen requirement. Those are the most important things. Next. Which of the following is not a gram-positive rod? Awesome job. Yep, Haemophilus influenzae, which does not cause the flu. The flu is caused by a virus. Which bacterium is the most common cause of urinary tract infections? So yes, this one's a bit of a tricky one. So E. coli is actually the most common cause of bacterial tract infections. We always think of it as it's causing GI tract issues like gastrointestinal problems, but it is also the most common cause of urinary tract infections, which is weird, but yeah, it can do lots of things. Next. All right, what is the definition of a nosocomial infection? Yep, an infection that is required in a hospital, which is why if you are staying in a hospital, you shouldn't stay longer than you need to. Usually they're through catheters. All right, which two bacterial genera are obligate intracellular parasites? Can't talk today. Yep, Rickettsia and Chlamydia. Yeah, Rickettsia and Chlamydia, that's an important thing to know. Just know which two genera of bacteria are the obligate intracellular parasites, Rickettsia and Chlamydia. They're the only two. What is the O2 requirement of bacteria in the genus Fusobacterium? It is a strict anaerobe. So not a facultative anaerobe, it is a strict anaerobe. It cannot have oxygen. So yes, good to remember as well. Know the oxygen requirements of the bacteria. Which of the following is an endospore producing aerobic gram-positive rod? Mhm. It is Bacillus anthracis. So this is an example of one of those questions. It involves a fact about it. It produces endospores. It involves its oxygen requirement. It has its stain and it has its shape. No, like each of those four things about each bacteria. They're the important things to remember. All right, true or false? Antiseptics are disinfectants that do not destroy living tissues. True or false? True. Yep. Antiseptics don't destroy living tissues, which is good because we use them on our skin sometimes. What does it mean if something is bacteriostatic? Yes, very good job, guys. No wrong answers there. Yep, if something's bacteriostatic, it inhibits the growth of microbes, but it does not kill them. Bactericidal kills microbes. Which of the following is not an example of a physical method to control microbial growth? Mhm. Yep, that is a chemical method, not a physical. Dry heat, osmotic pressure, and desiccation are all physical methods of controlling microbial growth. But gaseous King sterilizers, they are a chemical method. All right. How does osmotic pressure work to inhibit the growth of microbes? This one's an interesting one. A hypertonic environment is created. Very good job. Yes. So osmotic pressure is uh, just making the environment so that the water wants to flow out of the cell. So hypertonic, remember cell shrivels up, looks kind of like an R, hypertonic. It's how I remember them. Hypotonic, it swells up with water, so it becomes an O. Hypertonic, it shrivels up, so it looks like an R. But yeah, just makes it makes a hypertonic environment so that the cell cannot, all the water flows out of it and it dies. Next. Blank are the most difficult microbes to destroy, and blank are the easiest microbes to destroy. Yep, it is prions and viruses with envelopes. So a bit of a tricky one as well. On your control of microbial growth PowerPoint, there is a slide that has the order that things are most difficult to kill to least difficult to kill. Find a way to memorize that and write it on a piece of scratch paper, write it over and over again. It's an important thing to remember, like what things are easier to kill than others. But yes, prions are the hardest thing to kill. But strangely enough, viruses with envelopes are easier to destroy than viruses without envelopes. Which of the following is not an example of a chemical used in chemical control of microbial growth? All right, yeah, it is radiation. Radiation is an example of physical control of microbial growth. Heavy metals do count as a chemical control of microbial growth. Heavy metal chemicals are used. So yes, heavy metals is an example of chemical control, but radiation is an example of physical control. Mhm. Which of the following best describes fungi? Mhm. Eukaryotic chemoheterotrophs. So fungi are not prokaryotes. They are eukaryotes. Even though they can be single-celled, that is still a single eukaryotic cell. They have organelles, and they are chemoheterotrophs. They require their they require organic compounds from the environment. They can't make them themselves. They're not plants. They are a completely their own thing. All right. Which of the following is not a characteristic of septate hyphae? 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 I never know how to say that word. Mhm. Hyphae grow by elongating at the tips. Yep, that's not a characteristic of septate. That is um, the rest of them are. So if it's septate hyphae, they, like the hyphae, are divided into uninucleate units, distinct chains, and have pores. Meeting will end in 10 minutes. Great. All right, we have plenty of time, and we can start another meeting after this too, to talk about test taking tips and things. What does it mean for a fungus to be dimorphic? Yep, it can exist as yeast or a mold. It can be unicellular or multicellular. Yeasts are unicellular fungi. Molds are multicellular fungi. Um, yeah. True or false? Vegetated mycelium grows in the air above the substrate. Yep. It is false. So vegetative mycelium grows in the air above the substrate. No. Sorry. I'm blanking on the other name. All right. This is why I have notes next to me.
Isn't it aerial?
Thank you. Yes, it is 100%. So aerial grows in the air above the substrate. It can produce spores. Vegetative mycelium grows on the fungus, not in the air above it. Thank you. Whoever said that, I don't have my notes open to fungus. Which of the following is a characteristic of asexual fungus spore type? Arthrocandida. So in this type, it is formed by fragmentation of septate hyphae into single cells. The other one, so the one that is produced in chains at the end of a candida spore, is, I have them right here. Nope. Yes, so, well, okay. Arthrocandida formed by fragmentation of septate hyphae into single cells. And then rounding and enlargement, that is Chlamydocondia. And then Blastocandida is a bud coming off of a parent cell. And then the last one, I can't find it, but Oh, yeah, no, sorry, it's a candida. Yeah. My brain just turned off for a second. I'm back. Which of the following is the correct order for sexual fungal spore formation? So there are 3 steps for sexual fungal spore formation. What is the correct order for them? Yep. So know the steps. It is plasmogamy, charogamy, and then meiosis. So yes, orange is the correct answer. Know those. So go over the steps and what happens in each step, and know the order that it happens. Okay, only a couple more questions. Which type of medically important fungi is an obligate intracellular parasite often seen in AIDS patients? It is Microsporidia. So yeah, really interesting thing. Obligate intracellular parasite. Not Zygomycota. Zygomycota is black mold. So it is medically important, but it is different, not an obligate intracellular parasite. And last question. Which of the following is the only mycosis that is contagious and can spread from person to person? Yep, cutaneous. The rest of them people can get, but they can't spread person to person. So the only fungal infection that can be spread person to person is cutaneous. And I believe that is the end of the Kahoot. So congratulations, everyone. Awesome job. Congratulations, Megan. All right. Great job, everyone. I'm going to stop sharing my screen now. And how are we feeling after the Kahoot?
We need to work a little bit on fungi.
Yeah.
Yeah.
I think I need to work on fungi too. Not gonna lie. But yeah. Yes. Yeah. No, fungi is definitely, everything else seems so much more like there's more information. There's not a lot of information in the fungi unit. And so I saved it for last too, but there is important information in it. It is a short one to study, but definitely like block out an hour to just know the key concepts in fungi. Okay, can I write in the chat or in the email about what to remember for bacteria hacks? Yes, and I can go over those as well. Either in the next one we jump all back on after this kicks us out in 3 minutes and 30 seconds. I can go over test taking tips and studying tips that I used for this unit specifically. Some stuff. Yeah, no, for stuff that's going to be on the exam, mostly follow her study guide. Her study guide includes the most important information. Like there are some things on the PowerPoint slides that are skipped over in class that are interesting information to know in the context of things, but aren't part of the big main concepts and probably won't be on the exam. So the study guide is definitely the best thing to go off of. Do I know how many questions are on the exam? No, I do not. I unfortunately do not help make the exam, so not sure how many questions will be on it. Are we able to take the exam anywhere, like online at home on Respondus? Yes, I believe all of your tests are, so it'll be just like the last one. It'll be, yeah, as long as your Wi-Fi is fine, exactly. Have somewhere that is a quiet environment that you know you can focus, where there's no other people. Like, so a study room or your room, if you have a roommate, maybe try to find a study room. Yeah, but make sure your Wi-Fi is good. I remember one time in the dorms, a fire alarm went off like right as I was finishing up one of these exams. I was like, oh no. And I might have actually finished the exam first because I only had 2 questions left and people like don't know how to cook mac and cheese in the dorms and the fire alarm goes off constantly. All right, 2 minutes. Any other questions? And we will sign on to another Zoom and I'll record that one as well to go over different study tips and different test taking tips. But yeah, I will also send out this Kahoot, or the Kahoot we just did, so you guys can practice it again on your own. And I will send out in another email just important like study test taking tips to know as well. Can you give tips on how to remember the gram-negative rods? It's the one group you haven't touched yet. All right. So the way I studied bacteria for this exam, I made flashcards of all of the ones on the study guide. So like all of the important ones on the study guide. And the flashcards had like a fact about them, their oxygen requirement, their shape, and their gram stain. And then I sorted them. I like had the physical flashcards, like having a physical like visual for me helped. And so first I was like, okay, I'm going to sort them by shape. So I sorted them. I tried to sort them like spirochetes here, cocci here, rods here, and saw how many were in each section. And then I was like, okay, now I'm going to sort them by gram stain. And so sorted them like that. Yeah. Yeah, I've been using flashcards, but there's so much that are in the study guide. Focus mainly on the bacteria that are on the study guides. And then if you have time, go over some of the bacteria that aren't. Can't stay for the next part of the view. Totally okay, Bella. Hope you have a great rest of your day. And here, it's going to boot us in like 2 seconds. So we'll start another Zoom and record that one as well. I'm going to stop recording now.